everyone, welcome to my channel Pure Biology, and today I come with another video that is related to the muscle contraction process or role of the actin and myosin filament. Okay, and what is the actin and myosin filament? That means their structures I have already uploaded in my previous my channel, and so you can just check out the structure of the actin and the myosin filament. Okay. And this is the topic, what is the function of the actin and myosin. I will try to explain. And before going to this topic, I want to say thank you to all of you who have already subscribed my channel, and please share this channel with your friends if this is helpful, and please give one like for each video. Try to give like because it will give me an inspiration for doing the work properly. Okay. So let's continue. What is the biochemical events occurring during one cycle of muscle? So first of all, what happened? The signal that is coming from the nerve. Okay, the signals that is coming from the nerve, and that triggers an action potential in the sarcolemma. Okay. So what is sarcolemma? There are many terms. I will just explain not here, because here I just try to explain the actual events occurring in the muscle contraction. So many terms are there. I have totally explained in my previous video, so you just check out every terms. Okay, here I will try to only explain the muscle contraction process or the function of the actin and myosin filament. Okay, so what is happening here? The nerve triggers the action potential in the sarcolemma, and the action potential that propagates through the T tubules. T tubules is the transverse tubules. So first the nerve triggers an action potential, and this action potential propagates through these tubules, and these tubules extend inward from the sarcolemma around each myofibril. That means you can say that these extend all over the myofibril. Okay. So T tubule that contains voltage sensitive proteins. Okay, these are the voltage, this T tubule that consisting of the voltage sensitive proteins, and these are activated by incoming action potential. So if the action potential is not coming by the triggering of the nerves, so here the proteins are not activated, they remain in normal condition. Okay. So activated these proteins trigger the opening of the calcium channel. So what is happening? First nerve triggers the sarcolemma, and this action potential that propagates through the T tubules, and this activated the voltage sensitive protein that present within the T tubule membrane. And what happening after that? The activated proteins that try to open the calcium releasing channels. Okay, so calcium releasing channels are opened by the activation of the voltage sensitive proteins, and in the sarcoplasmic reticulum. So these are the channels that are present in the sarcoplasmic reticulum. Just like the endoplasmic reticulum, sarcoplasmic reticulum is present in the muscle cell. So increase in the cytosolic calcium. When the calcium level increase, the contraction of each myofibril try to occur. Okay. So when the calcium, cytosolic calcium level that becomes increased, then the contraction of the myofibril starts. Okay. Because the signal from the sarcolemma, this is passed within millisecond to every sarcomere, and what is saying that within millisecond the signal that spread all over the muscle cell. That's why the myofibril in the cell contract at the same time. Okay. The increase in the calcium concentration is transient. Why this calcium concentration is very important? Because this calcium is rapidly pumped back into the sarcoplasmic reticulum and by the abundant calcium ATPase in its membrane. Typically, this cytoplasmic calcium concentration is restored to the resting level within 30 milliseconds, allowing the myofibrils to relax. What is saying that? Again, I repeat this line. The calcium concentration increase is because the calcium is rapidly pumped back into the sarcoplasmic reticulum. Okay, calcium is pumped back into the sarcoplasmic reticulum by what? By the calcium ATPase that present in the membrane, and that's why the calcium level or the concentration of the calcium that become increased. Clear? And now this calcium concentration is restored to the resting level within 30 milliseconds, and this allowing the myofibril relax. When the calcium concentration not changed, that means this is called the resting level. You all I think I never explained about the action potential and the resting level, so I will try to explain this action potential and resting level in when we learn about the nerve cell. Okay, so you just learned here the relax means the action potential is not there, and resting phase means when the action potential is not there. Action potential when occurring when the nerve triggers. Okay, when the nerve triggers, then action potential occurring, and otherwise the relaxing phase or the resting phase is seen in the muscle cell. Clear? So what is saying that this resting level, when the calcium concentration that restored in the normal situation, then the resting level is seen and the myofibrils remain relaxed condition. And the calcium dependent skeletal muscle contraction is due to the presence of the tropomyosin and troponin. So two important term is very needed for the muscle contraction, because only calcium cannot important. It is not only one that control the muscle contraction. Two important terms. One is the tropomyosin and tropomyosin. These are the two important terms that are important for the muscle contraction. With the obviously with the calcium concentration, but these two terms are very important. So each tropomyosin that present where along the groove of the thin filaments. Okay, there are thick filaments present and the thin filament present in the muscle cell or muscle fibrils. So thin filaments, there are the grooves. So these grooves that contain the tropomyosin. And in case of resting muscle, the troponin 1 and troponin T interact with the tropomyosin to keep tropomyosin away from the groove of the thin filament. So what is saying that when the troponin 1 and troponin 2 both are connected, then they connected with the tropomyosin and keep the tropomyosin away from this thin filament. Clear? That means tropomyosin when comes in the thin filaments, then muscle contractions may be possible. And this when the tropomyosin just remain away from the groove of the thin filaments, what is happening? The binding of the myosin heads with the actin filaments that is prevented. Okay, binding of the actin and myosin, these are prevented. And so why this is prevented? Because the tropomyosin not remain within the groove of the thin filaments. These are away from the thin filaments. Okay, so the binding of the myosin and the actin filament is not possible, and that's why no force generating interaction occur. No force generating interaction occur. When the actin and myosin both are binding with each other, then the force, any force that is necessary for the muscle contraction, this is occur or this is formed. And when the calcium level is raised or increased, then troponin C, which binds up to 4 molecules of the calcium, and that causes troponin 1 to release and hold on the actin. Okay, so what is saying that when calcium level increased, then calcium level when increased, this calcium causes troponin 1 to release the actin filament. Okay, and this allows the tropomyosin molecules to slip back into their normal position so that the myosin heads can walk along the actin filament. What is saying that? Again I repeat, when calcium level that is rising, then troponin 1 release to release its hold on actin, and that allows the tropomyosin molecules to slip back into where? Into their normal position so that the myosin heads can walk along the actin filament. So calcium level if not increased, so the myosin and actin filaments interaction is not possible. Clear? So this is the biochemical events that occurring during one cycle of the muscle. Next part, I will try to explain about the coupling of the actin myosin and the ATP hydrolysis. Okay. So this is the another topic. What is called the coupling, and this topic is generally ended here. Okay. So continue with my channel.